QIMR is definitely recognised globally for its contribution to liver disease. My father, Professor Laurie Powell, was our director of QIMR Berghofer. He was the head of a liver program of research, as well as an active clinician. It's probably only now, as a more senior clinician, that I recognise really what a major contribution he made. One of the comments, I remember he had many phrases, many comments to us as children was, a day without an experiment is a day wasted. So he was a passionate researcher.
One of the fascinating things about medical science, of course, is the more questions you ask and answer, the more questions emerge that have to be answered.
The partnerships between clinicians and scientists, Dad thought, were vital to the success of a research program. I'm delighted to have the privilege to continue to promote engagement between clinicians and scientists, to continue a program of work that my father developed or initiated those many years ago. It's very special, and it's a privilege to be able to continue that relationship.
I feel very fortunate to have been employed in the first place by June Halliday and Laurie Powell, and to have them move to QIMR in 1990, because I was part of a generation of researchers who made a real difference to patients with this inherited iron overload disease, haemochromatosis.
And that disease produces masses of iron in the liver, far too much for the body to deal with, produces scarring, which leads to what's called cirrhosis, which you may have heard of. And ultimately, about a third of those people, if they're not diagnosed, will die of liver cancer. So it's a major problem.
June did have a long history with the Institute as a young woman scientist.
June was an incredibly strong, determined investigator. Laurie used to breeze into lab meetings wearing his flying white lab coat. He was so excited to hear the results and hear our theories and what we were going to do with them. They did an enormous amount of work to try and narrow down the region of the chromosome where the genetic defect for haemochromatosis was found. They didn't eventually identify the gene, but the group that did had enormous resources and used all the information that was published by our liver unit at the time. So you can now be tested, a genetic test, to identify whether you have a mutation in the HFE gene for haemochromatosis. 1 in 200 Australians of Northern European descent will have that mutation, so they can be diagnosed much earlier and they can get treatment, which is simple blood donation, and they can have a perfectly normal life. And if it wasn't for the research that was conducted at QIMR, we wouldn't be able to do that now.
The work we've done at QIMR Berghofer has contributed in a large way to that pool of discoveries that have led to a greater understanding. I think the two major discoveries that we've generated, the first is the identification of a gene that is essential for efficient iron absorption from your diet, called hephaestin. So that it was a fundamental discovery. And the second was the demonstration that HFE, the gene affected in haemochromatosis, we now know that it controls this hormone that affects how much iron you absorb from your diet.
And that changed the whole paradigm of understanding how haemochromatosis works and potential future treatments. People at QIMR Berghofer who've worked on haemochromatosis feel like we've done an awful lot of research over the years in that disease, but there's always more work to be done. Over a period of 40 or 50 years, Professor Powell, through his normal clinical practice, put together a patient database. Once Professor Powell retired, we discussed what we would do with that database, and it was felt that we should share it. So that resource at Haemochromatosis National Registry is going to seed a lot more research. It's a phenomenal resource, one that I'm very proud that I was able to shepherd through to assist patients, also as a legacy for Laurie. We've been able to identify how scarring occurs in the liver, and we've been able to demonstrate new non-invasive tests to demonstrate that patients have serious scarring of the liver. Some of those markers are now being used clinically by doctors who can identify these patients have serious liver disease, but without subjecting them to a liver biopsy. So that's just a practical example of that resource in action.
There's so many questions and issues to address, you never can do that. So it's great that you have people who will at least continue the general theme. They might follow specific areas. And David at Fraser and Sheridan Hellman, they're taking some innovative approaches to try to diagnose and treat iron deficiency. And that work has the potential to help a lot of people. The research done at QIMR Berghofer is very important, and that's why we've been in business for 80 years now. And through that time, we've been one of the most influential research institutes in the country because we've understood the biology and translated that into patient outcomes.